The movement of materials in and out of a living cell involves many processes. Of these, perhaps the most basic and best understood is the physical process of diffusion. Diffusion depends on the motion of molecules. We can get evidence of molecular motion by observing a drop of milk under the microscope. Fat globules in the milk move back and forth at random. They're being bombarded by molecules, which themselves move about in a random fashion. To show the effect of the random movement of molecules, we let some colored gas into a closed container. There is a movement of molecules from an area of greater concentration to an area of lesser concentration. This is the process of diffusion. After a period of time, the gas is diffused evenly throughout the container. To show how diffusion is involved in the movement of materials in and out of a living cell, we'll represent a cell using this smaller plastic cube. The sides are of an extremely thin plastic, which functions as a membrane. This membrane has physical properties similar to the membranes that enclose living cells. Materials which enter or leave a living cell pass through a membrane which surrounds the cell. Let's see the effect of the membrane on the diffusion of a gas. With the cell inside the larger container, gas is admitted into the cell. At first, the gas seems confined within the membrane. As time goes on, the gas diffuses through the membrane. Diffusion continues until the concentrations of gas within and without the cell are equal. This helps us understand how gases such as oxygen and carbon dioxide diffuse in and out of living cells. All gaseous exchange in plants depends on the process of diffusion. Diffusion also accounts for the exchange of gases in animals. Oxygen and other gases in the air enter through the rabbit's nose and move into its lungs as the animal breathes. Inside the lungs, the gases move into clusters of air sacs called alveoli. In each alveolus, oxygen gas is more concentrated than in the surrounding capillary where the blood cells are. So oxygen diffuses into the blood. In the blood cells, carbon dioxide is more concentrated than in the alveolus. So carbon dioxide diffuses out of the blood cells. In much the same manner, gas diffuses in and out of other living cells. Living cells are composed primarily of water and generally exist in water solutions. Immersing the cell model in a container of water helps us simulate the physical conditions encountered by a living cell. In the water outside the cell membrane, we'll pour some molasses. Molasses contains glucose, one of the simple sugars used by living cells. We'll see if molasses will diffuse through the membrane. We'll time the diffusion process. At the start, the inside of the cell is clear. About 6 hours later, some of the molasses has penetrated the cell membrane. After 12 hours, the molasses concentrations outside and inside the cell are nearly equal. This membrane is permeable to molasses. Starch is another material used by living cells. We'll pour the starch into the water outside the cell as we did with the molasses. We'll see if the membrane is permeable to starch. 6 hours later, the starch has diffused throughout the surrounding water, but has not permeated the membrane. At the end of 12 hours, the starch still has not penetrated the membrane. So this membrane is impermeable to starch. A membrane that is permeable to some substances and impermeable to others is called a semi-permeable membrane. Semi-permeable membranes surround living cells. Such a membrane is generally permeable to such things as gases, water, sugars, and salts. It is generally impermeable to substances like starches, fats, and proteins. This semi-permeable quality of a living cell membrane helps explain why, in blood cells, gases readily diffuse in and out, while large protein molecules stay within the cells. In the small intestine, large fat molecules must be broken down into smaller glycerin and fatty acid molecules to permeate the membranes of the intestinal wall. In plant protoplasm, starch must be broken down to sugars to diffuse through the membranes surrounding the cells of the plant. All of this movement in and out of cells takes place in water. With this plastic sac, we'll investigate the diffusion of water through a membrane. The egg will provide protein material as found in a cell. Now the membrane is placed in water. See if you can predict what will happen. The sac swells. Inside the sac are water molecules and molecules of protein. Outside the sac are only molecules of water. Water, then, is more concentrated outside than inside, so water diffuses into the sac. The diffusion of water through a semi-permeable membrane is called osmosis. We can illustrate osmosis in a living cell with the aid of some red blood cells viewed under a microscope. The concentration of materials in the water outside the cells is almost the same as inside. There is osmotic balance. We can upset this balance by adding a drop of distilled water. Watch what happens to the blood cells now. They expand and burst. Water, moving into the cells by osmosis, creates osmotic pressure, enough to destroy the delicate membranes. Another example of osmosis can be shown using plant cells. We'll add a salt solution to some algae plants. The water molecules outside the cells are less concentrated than inside. In this case, water diffuses out of the cell, causing the membranes to shrink away from the cell walls. The protoplasm is crowded into the centers of the plant cells as a result of osmosis. Using another plant, we can demonstrate how osmosis is involved in absorption of water through roots. The plant is being placed into a concentrated salt solution. This will reverse the process of osmosis, removing water from the plant. In time-lapse photography, we see the plant wilting. We can restore the stiffness to the plant by removing it from the salt solution and placing the roots in fresh water. To show that the water diffuses back into the plant, we'll use time-lapse photography once again. Osmotic pressure against the cell walls causes a firmness known as turgor. The maintenance of turgor is an important effect of osmosis. Osmosis, remember, is a special case of diffusion. Diffusion of water through a semi-permeable membrane. The diffusion of water, the diffusion of glucose, the diffusion of gases and other materials helps account for the movement of materials in and out of living cells.